Good morning, everyone. Today, we will finish our chapter on the cytoskeleton. We'll look at the different components of muscle contraction, and then we move. Oops, I'm getting a message. My internet is unstable. Hold on. Did I not put on? Now I should be good. All right, I should be good now. Okay, I'm on my 5G. Makes a big difference. Uh, we'll look at muscle contraction. I'll show a little animation of the muscle contraction, and then we'll move on to our next chapter that will start with apoptosis and then look at cell renewal as well. So where we left off, we were describing the interaction of the filaments in that sarcomere. The myosin head starts by attaching to the actin filament. The myosin head then binds ATP. It hydrolyzes ATP. That causes a change in the shape of the myosin to the cocked position, which then, once the ADP and inorganic phosphate are released, results in another change, which is the power stroke, the movement of that myosin head, resulting in myosin filament walking along the actin filament, right, towards the plus end. All of those individual myosin heads moving simultaneously along the actin filament will cause the filaments to slide relative to each other. So what would happen if the cell ran out of ATP? What would happen to the interaction of the actin filaments and the myosin filaments? And that's our first question for today. The process that we're just looking at is the myosin head binds ATP, it hydrolyzes ATP, and then it releases those products of the hydrolysis, all of those individual shape changes resulting from interaction with ATP. So what happens if the muscle cells run out of ATP? Is one of the reasons why your muscle cells have so many mitochondria, to be able to generate a huge amount. I think it's something like 1,000,000 ATPs are consumed per minute or are needed per minute, maybe per second, 1,000,000 per second. That sounds more like it, in a muscle cell, in order to carry out this contraction process. How are we doing? About 5 more seconds? What would happen if the muscle cell ran out of ATP? All right, let me close it up. And what would happen is that the head would remain stuck, because that's the starting point, right? Remain stuck, attached to the actin filament. In the absence of ATP, there would be no change, right? The head would not be able to change its shape. By hydrolyzing the ATP, it would not move along the actin filament. And this is rigor mortis. This is the reason why when a body dies and can't generate any more ATP, all of the muscle cells are stuck in whatever point of contraction they were at. The myosin and filaments remain in sort of frozen in their position relative to each other. So our muscle cells need ATP in order for this myosin filament to walk along the actin filament. But what's regulating this contraction? Because muscle cells in a healthy, normal cell are not always contracted, right? That contraction is going to be stimulated by the motor neuron. And indirectly, the stimulation by the motor neuron is affecting the concentration of calcium in the cytosol. An increase in calcium is required for a muscle to be able to contract. And the reason for that is the actin filament is bound to some accessory proteins or regulatory proteins, one of which is this long rod-like protein that we call tropomyosin that's bound to the actin filament, sort of in that groove between the two helices, bound to the position where the myosin head would interact with actin. This tropomyosin is also bound to another regulatory protein that we call the troponin complex. And the troponin complex is sensitive to the concentration of calcium in the cytoplasm. So what we're going to see is how calcium interacts with troponin complex, which affects the positioning of the tropomyosin on the actin filament. In this position, the actin filament cannot bind myosin because the myosin binding sites are blocked or masked by the tropomyosin. What we're going to see is the troponin complex is a regulatory component. When it binds calcium, it will induce a rearrangement of the tropomyosin. So let's look at that on the next slide. What we've done here is we're looking at sort of a cross section of an actin filament. And over here is the myosin binding portion of an actin filament that we can see in the resting position is bound by a cross section of the tropomyosin. Over here, not shown, would be the troponin complex. In the presence of calcium, the troponin complex will change its shape, which results in a movement of the tropomyosin away from the myosin binding site, which then makes this actin filament able or capable of binding to myosin. Right? So the cell requires an increase in calcium in order for the actin's myosin binding site to be exposed. So what's controlling calcium ion concentration in the cell? And when we're talking about in the cell, what we really mean is calcium ion concentration in the cytosol, because this is where all of these myofilaments reside, in the cytoplasm itself, packed with these actin myosin filaments. Well, we described earlier, one of the roles of the smooth ER is to store calcium in all cells. Calcium is constantly being transported into the endoplasmic reticulum. The endoplasmic reticulum is, again, will have a high concentration of calcium, and in most cells, the cytosol has a low concentration of calcium. Muscle cells have a special type of ER that we call sarcoplasmic, sarco referring to muscle, which we can see on the next slide. Here we're looking at our myofibrils, all of those packed fibers of actin and myosin, completely surrounded by sheets of endoplasmic reticulum that we call sarcoplasmic reticulum, looking at muscle cells. And we also see that the membrane of the sarcoplasmic reticulum, which is storing high concentration of calcium, is very closely associated with the plasma membrane, because there are some invaginations of the plasma membrane called T-tubules, making sure that all regions of the sarcoplasmic reticulum are, at least at some point, closely, even when they're wrapping around internally, are closely associated physically next to portions of the membrane. So if we sort of take a cross section through this, or I guess a longitudinal section through this, we would see that our T tubules, a cross section through a T tubule, which is really continuous with the plasma membrane, is very closely, physically close to regions of sarcoplasmic reticulum membrane. And we're going to see why that's important, because changes to the plasma membrane are continuous with changes in the T tubules, and that's going to affect the membrane of the sarcoplasmic reticulum. So again, thinking what's going to induce a change in shape of the tropomyosin that's going to allow for contraction. We know it's initiated by a signal from a motor neuron, and a motor neuron interacts with the muscle cell at the membrane, right, at the plasma membrane. And so the sequence of events is initiated by a depolarization event. The motor neuron interacts with the membrane of the muscle cell, depolarizing the plasma membrane, which therefore depolarizes the T tubules since they're continuous with each other. And then we're going to look at what happens when a T tubule membrane is depolarized. That's going to result in opening of a voltage-gated calcium ion channel, which will activate something even more internal to that. So let's start from the beginning sort of, and then follow through what's going to result in sarcomere contraction. We remember that at a neuromuscular junction, when the electrical signal reaches the nerve terminal, causes fusion of the secretory vesicle, releasing the neurotransmitter. Remember what that neurotransmitter is? Anybody remember? Acetylcholine, right? Acetylcholine will then bind to receptors on our target cell, which is our muscle cell. That receptor is a sodium ion channel. Binding of the acetylcholine causes the sodium ion channel to open. Sodium ions enter. That changes the membrane potential of this muscle cell. What we're going to see is what happens downstream of that. What's the consequence of that depolarization of the muscle cell membrane? Because there are some voltage-gated calcium ion channels on that plasma membrane. And really what we're looking at, remember, our plasma membrane is invaginated into the interior of the muscle cells, and that's what we're going to look at on our next slide. Over here on the left is at rest, we have our T tubule, which is continuous with the plasma membrane. We have a voltage-gated calcium ion channel in the T tubule. Physically next to the membrane of our sarcoplasmic reticulum. No calcium, or relatively low concentration of calcium in the cytosol. When the plasma membrane is depolarized by receiving that signal from the motor neuron, that results in depolarization of the T tubule membrane, which results in the opening of the calcium ion channel, letting into the cell just a tiny bit of calcium, which binds to a channel on the surface of the sarcoplasmic reticulum. That channel will then be opened, which will result in a burst of calcium ions, a huge increase in calcium ion concentration in the cytosol, which will result in binding to the troponin complex, changing the shape of the troponin complex, which results in changing the shape of the tropomyosin, which will then unmask the myosin binding sites on the actin filament, allowing for the beginning of contraction. Right, it's very quick. All of the sarcomeres will be able to contract at the same time. And then when that signal from the motor neuron ends, all of the myosin will release the actin filaments because sort of in the background, calcium is constantly being transported back into the sarcoplasmic reticulum. So it's a quick, short burst of calcium ion increase causes the contraction. The absence of that calcium increase results in relaxation of that sarcomere. Right? A lot of pieces we have to put together and think of how quickly that happens. So let's take a quick peek at a very nice animation that puts all of these together, sort of the same thing I've been talking about, but puts it together sort of from beginning to end. Let's close that down for a second. Open up this guy.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads from the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction. In each contracting unit, or sarcomere, thin actin and thick myosin filaments are juxtaposed, but cannot interact in the absence of calcium. This is because myosin binding sites on the actin filaments are all covered by a rod-shaped protein called tropomyosin. A calcium-sensitive complex, called troponin, is attached to the end of each tropomyosin molecule. When calcium floods the cell, troponin binds to it, moving tropomyosin off the myosin binding sites. Opening the myosin binding site on the actin filaments allows the myosin motors to crawl along the actin, resulting in a contraction of the muscle fiber. Calcium is then quickly returned to the sarcoplasmic reticulum by the action of a calcium pump. Without calcium, myosin releases actin, and the filaments slide back to their original positions.
There we go. So how is this regulated? Let's think about this here. So which of these conditions would decrease the likelihood of a contraction?
And while you're answering this, you might want to think about, would you be able to draw that sequence of events, starting with the motor neuron resulting in contraction or movement of actin filaments relative to myosin filaments? Would you be able to draw sort of that flow of information or signal and regulation from motor neuron to the contraction? That would be a good way for you to test your understanding of all of the different steps in this process. All right, how's everybody doing? So we're looking for which event would decrease the likelihood of contraction. 5 more seconds. All right, let me close it up. All right, let's see what we've got here. It looks like the popular answer was D. I saw that a couple of people were worried that they didn't see the previous question. They didn't see the answer chosen. And every once in a while, I click too fast and it's not here. But I always double check when I'm putting up points. I always check that I have selected the correct answer on the questions that I grade. Remember, I don't grade every one of them, but the ones that I'm grading, I always check them before I put up the points. All right, so if I'm not paying attention to the actual grading during class, I do pay attention to it before the grades are posted. So D is the correct answer here. D is the only one that would decrease contraction, right? If we're depolarizing the T-tubule membrane, anything that's even making the causing even a slight depolarization would be increasing the probability of a contraction. If we add more ATP, we're going to increase the probability of a contraction. Or if we interfere with tropomyosin's binding to that actin filament, then that myosin binding site on the actin filament will remain available. And so, but a drug that will block the binding of the calcium, even if the calcium concentration is high, blocking the binding or the ability of the calcium to bind to troponin would prevent the downstream events that would lead to contraction. Okay. Right, and I really think that that animation is an example of something that really puts all the pieces together very nicely to help you see the sequence of events. All right, so that concludes our chapter on our cytoskeletal components. Make sure that you know what are the roles of these different types of cytoskeletal filaments. Some of them are stable, or in some circumstances, some filaments remain as a polymer. Others are growing and shrinking. What's the difference, and what's the function of those different types of stability? What's the role of motor proteins in moving vesicles and organelles around the cell? What's the role of a motor protein in movement of cellular extensions like cilia and flagella? What's the role of motor proteins in muscle contractions? Think about how you could describe what's causing the motor protein to move, which motor protein interacts with which cytoskeletal filament, right, to carry out these different types of roles. So I'm going to close this one down, and we're going to move on to a different topic in our cells. We're going to move on to apoptosis. In this chapter, I'm combining concepts from both the process of cell death as well as the process of cell renewal, right? Homeostasis, both contribute to homeostasis of a multicellular organism. That organism maintaining the correct number of cells that are needed for the tissues to function, in collections of tissues and organs. So we're first going to look at what is this process of apoptosis, this active process, active death. And then later we'll look at where are the cells coming from to replace dead or dying or damaged cells in an organism, or even during growth. So we saw this figure earlier, so how cells, when we're talking about cell communication, cells need to receive signals to change their behavior, to grow, to divide, to differentiate. But even just to survive, cells have to receive signals just to stay alive. In the absence of signals, a cell will undergo apoptosis, this programmed as in genetically programmed cell death. So you can really think of cell death as being the default state for many cell types. This And what our cell, by receiving survival signals, will have to do is sort of override that default state. In other cases, an infection or damage to a cell or damage to cell components will induce apoptosis. So we'll look at a couple of different examples as to why cells undergo this programmed cell death, and then we'll look at the process. How does that actually happen? So a normal process in normal development does involve both cell division, cell differentiation, but also cell death. Over here, we're looking at the development of a fetal mouse, development of its paw. Early in fetal stage, it's sort of spade-like. And if we look closely, we see there are some fluorescently labeled cells. These little yellow dots are cells that are undergoing apoptosis, regulated cell death. The death of those cells results in, a day later, the sculpting of individual digits of this paw. Right? So this is a normal and necessary event in normal development. Another example of apoptosis in a regulated, in this case, a hormone regulated cell death during metamorphosis, right? An amphibian that has one part of its life cycle, a tail necessary for locomotion when it's living in its water environment. When it metamorphoses into an adult frog and it has a terrestrial lifestyle, it doesn't need its tail anymore. And so that tail, it doesn't just fall off, that tail slowly regresses over time by slow regulated death, apoptosis of those individual cells. Right, so cells need to remain alive if they have a function. If the cell, once those cells have carried out their function, if they're not needed anymore, then they're removed by this type of process. So what is apoptosis? We're saying that it's energy dependent, meaning this is an active process. It's not passive death. It's not necrosis. It's a programmed cell death. Can be triggered by damage to the DNA, by a viral infection, but it can also be, those would be considered, let's say viral infection could be an external signal, but it can also be initiated by internal cues, a developmental program like what we saw in the case of metamorphosis, or in the case of the development of the digits on that mouse paw. And we'll look at what some of those cues are, internal signals or external signals. No matter what cell type we're talking about, and no matter what is sort of the initiating event, apoptosis usually involves this sequence of events. Starting with fragmentation of the DNA, meaning the DNA is broken up into little bits. This is caused by activation of a DNase, a pro-apoptotic, pro meaning promoting, a specific DNase that's going to lead to cell death. That results in, the fragmentation of the DNA results in chromatin condensation. And when we say condensation here, we're talking about sort of unspecified precipitation, right? The chromatin is just breaking down into and shrinking up. Not normal condensation as we would see during, as the cell's preparing for mitosis or cell division. We'll also see the nucleus breaking down. And again, that's not normal mitotic, regulated, temporary breakdown of the nuclear envelope. It is destruction of the nuclear envelope by the activation of pro-apoptotic proteases that target the nuclear lamin. So the DNA is precipitating, the nucleus is breaking down, and then other proteases, and a protease is always an enzyme, right, that targets proteins, right? So it's going to cleave peptide bonds in proteins. Some pro-apoptotic proteases will be targeting cytoskeletal proteins. So the cell's breaking down. It's fragmenting into these small condensed bodies that we call apoptotic bodies. Now, again, we want to differentiate this series of events, these regulated events, from another type of cell death, which is necrosis. Necrosis is when it's sort of an accidental death, when a cell is damaged, it swells, it lyses, it releases its contents, it then causes inflammation by surrounding cells. It's a messy death. In contrast, apoptosis, which is an active process, is a controlled and neat death, where all of the cellular components condense and shrink up, and then will be phagocytized by a surrounding cell. Another cell will recognize that this is a dying cell and will take it up, right? Will phagocytize it and then recycle those components. So the cell's gone, but there's no damage to the surrounding cells. And in fact, all of its matter, right, will be recycled by those surrounding cells. So one question then is, how does this phagocytic cell, this neighboring cell, recognize that the cell within its cellular context, how does it know that that this cell needs to be removed? Well, the other thing that this apoptotic cell does is it expresses a signal on its surface. What it's expressing on its surface is a phospholipid. A phospholipid, for example, phosphatidylserine, which is normally positioned on the inner layer of the lipid bilayer, usually facing the cytoplasmic side, that will be actively flipped to the outside, right? An enzyme will move that phospholipid so that it faces the extracellular side. And that serves as an eat me signal, signaling surrounding cells to phagocytose it. And that's what we can see here. On a normal cell, if we look at our lipid bilayer, we would see that phosphatidylserine faces the cytoplasmic side. And remember how we said that phospholipids can move laterally, right, within a layer, but they can't move, they can't cross that, that hydrophobic barrier, except when an active process, an enzyme, will specifically move them, and this is part of the apoptotic cycle or process, move that phosphatidylserine so it's facing the outside, it's in the extracellular layer of the lipid bilayer, where it can be bound by, recognized by receptors, right, that are present on that surrounding cell. Right, that will activate that phagocytosis. The surrounding cell, the neighboring cell, will wrap around and then take that cell in, pass it on to a lysosome, which will then, in a regulated manner, degrade. And this phagocytic cell can then utilize those components to synthesize its own requirements. All right, yes, that's flippase, correct. Yes, it is. So what do we know about, what can we say so far then about, without knowing who's actually doing this, can we say about programmed cell death? And we'll talk about some of the signals in a couple of minutes too. How are we doing? About 5 more seconds? We'll talk about that in a minute. Okay, we'll talk about the signals. All right, everybody have a chance to vote? All right, let's close it up. Yes, this program, cell death, the only statement that describes it correctly, it's an intracellular suicide program. The cell is activating a sequence of events that results in this nice, neat breakdown and condensation of components. It occurs in normal development. It occurs frequently. It can occur in unhealthy, damaged, abnormal cells, and can occur at any time in development, as well as in adult tissues as well. In plants, it's a similar type of cell death occurs, similar, but because of the cell wall and because of different and the absence of like an immune system that would recognize dying cells, it's called senescence in plants, a similar type of cell suicide program involving the same kinds of actors, and that's what we're going to talk about. Yeah, what's actually carrying out this? What are these proteases, right, that are breaking down in this regulated manner all of these different cellular structures? Well, this was first discovered about 35 years ago by geneticists studying C. elegans, a free living nematode, a soil nematode, lives in freshwater, but can also be cultured in a lab. They're really amazing because they're so tiny that you can grow thousands of them on a petri dish. They are very simple. They only have a few different cell types. They're transparent. And because of all of those features, this organism we can follow every single cell from that first zygote, every single cell division, follow the cell division as well as death of individual cells. An adult worm has exactly 959 somatic cells, plus, of course, a varying number of germ cells. During its development, exactly 1,090 cells are formed, but 131 of them are eliminated at different stages of the development of this animal. And that can be followed, has been, by tagging individual cells, following them along the way, to be able to construct this developmental history, starting with a single-celled zygote. When it divides in two, this cell gives rise to two more cells, and this cell gives rise to two more, and you can follow the final destination of every single cell, making up primarily digestive systems. These are pretty simple. They have skin, they have digestive system, they have some neurons, and then they have, once they've molted 1, 2, 3 times as an adult, then they have reproductive system. So they're very simple to follow. Every single cell, when it divides, when it differentiates, and when it dies, if and when it dies, can be followed. You will not be tested on those individual cells, but because of being able to follow the cells, this is the kind of thing that the geneticists were able to discover. If you look at a C. elegans animal, and this is probably a larval stage, what you see is occasionally some specific cells are undergoing death. They're dying, and you can tell the dying cell by exposing the animal to polarized light, because the dying cells will refract or reflect the polarized light differently from living cells. All right, so that's a normal dying cells, which at first, when they first discovered it, was like, that seems very strange. Why are cells dying? We didn't know that this goes on in all organisms. So in order to study a process, that one way to study is to create mutations, right? Interfere with the process. And so they generated a huge number of mutations, some of which resulted in the absence. So where there normally would be 3 cells in this region of the animal dying, in this particular mutant that we call SED3, which stands for cell death 3, those same cells were surviving. And so they generated and screened for and found a number of cell death mutants, which they could then, using reverse genetics, determine what is the protein that's responsible for the normal process, right? What are these cell death genes? And then what they discovered is, for example, cell death 3, SED3, the gene that has been mutated in that animal is a protease, a pro-apoptotic protease, meaning that the SED3 gene product is required for cell death. Its activity results in cell death. SED4, another mutant, when we determine what its gene product is, the normal SED4 is required to activate SED3. And so genetically, if you have a null mutation, null means no gene product, right? The absence of any gene product from either SED3 or SED4 leads to survival of cells that would normally die. Right? It's meaning that the normal gene product is required for cell death. And then they found another cell death gene, and that appears to be an inhibitor. SED9 appears to inhibit, that's what our little bar is, right? Inhibits the activity of SED4. So a mutation in SED9 would lead to cell death. The absence of the inhibitor allows SED4 to activate SED3, which results in SED death. The counter to that is a mutation that results in overexpression of SED9 or overactivation. So excessive inhibition by SED9 leads to survival of cells that would normally die. Right? So this is an example of where genetics, by mutating, screening for collections of animals that have mutated different components, helps you piece together what are the normal sequence of events. So we've got an inhibitor and we've got a couple of activators of cell death. So what are these, right? So let's look at SED3 specifically. What kind of protease is that, and how does it carry out this cell death? SED3 is a caspase, a particular type of protease. We call it a caspase. The C for cysteine, the ASP for aspartic acid. This is one component of a number of caspases. So we, as multicellular organisms, produce a number of caspase proteases. They are the executors. They carry out apoptosis. The reason for the name, the C, the cysteine, is part of the protease that in the active site that actually interacts with the target protein to carry out the hydrolysis, the peptide bond hydrolysis. So that's where the cysteine is, in the active site of the protease. And the aspartic acid refers to the amino acid in the target protein that's sort of attacked. All right, we'll see an example of that on the next slide. So caspases target different proteins in the cell. Some of the targets are, for example, one target is an inhibitor of a DNase. Now, DNase is an enzyme that digests DNA. So if a caspase cleaves and destroys the inhibitor, then that means the DNase is free to break down and destroy DNA. We'll see an example of that in a second. Another target of a caspase are cytoskeletal proteins. Cleave those cytoskeletal filaments, causing all of the structure of the cytoplasm to break down. Another target is a nuclear lamin. So let's look at this one in a little bit more detail on the next slide. The action of a caspase on a nuclear lamin resulting in permanent breakdown of the nuclear envelope. Excuse me. Now we know from our previous, we've discussed this a couple of different times, that our nuclear envelope is lined on the internal membrane by our lamina, right? These are type of intermediate filaments. And each of those lamin components are this globular rod shape, right? We've got this dimer that interacts with another dimer. We have tetramers, and then we have these 8 filaments that all wrap around to make this very strong cable of an intermediate filament, our nuclear lamina. Well, in the middle of the rod portion of the individual subunit is an amino acid sequence, which constitutes a caspase cleavage site. It's the target site for a caspase protease cleavage. D is the single letter abbreviation for aspartic acid. So the peptide bond just after that aspartic acid, between the aspartic acid and this asparagine, will be cleaved in every single lamin subunit, which will break down, break down the entire nuclear lamina permanently, irreversibly. Remember, we said that during mitosis, the nuclear envelope will temporarily sort of fall apart by phosphorylation, but the phosphorylation can be reversed and the envelope can then come back together at the end of mitosis. This type of action will permanently break down, right? Nuclear lamina will not be able to reform again. And so that's one of the sort of downstream actions of a caspase to cause breakdown of the components of the cell. Another target of a caspase is an inhibitor of a DNase. So if the caspase destroys the inhibitor, then the DNase is activated. It's a pro-apoptotic DNase. There's lots of different DNases in the cell, but this is one that will target all of the DNA that is not protected by being sort of wrapped around the nucleosome core particle. So the target of this DNase is everything. It's all of the DNA in between all nucleosomes. And you can actually see that, you can see a dying cell in this image of DNA run on a gel, on an agarose gel. In lane 2, we have this very high molecular weight of chromosomal DNA. In lane 3, we're looking at the DNA from a cell that's dying. Yes, some of the DNA is still high molecular weight, but we're starting to see sort of a ladder of fragments that are 200 base pair difference in size, because the distance between one nucleosome and another is about what the amount of DNA that's wrapped around that particle is about 200 base pairs. So we'll see there's 200 and a 400 and 600 and 800. This sort of regularly spaced DNA fragments that are characteristic of a chromosome being fragmented, right? Another target of a caspase. So the caspase is the executor. The caspases, these are the proteins that are causing all of the outcome of that's leading to cell death. So one question then is, how come all cells are not dying all the time? Because all cells produce caspases, but all cells, all of your cells that are alive right now and functioning, are producing an inactive proenzyme, an inactive form of a caspase that we call a procaspase. A procaspase has to be activated to function as a caspase. It has to be cleaved, right? It's activated by proteolytic cleavage. So that's what we see here. All of our cells have procaspases, but not all of our cells have our procaspases activated. So if we look at a cartoon of a procaspase, we have sort of 3 different regions. When it's activated, it will be cleaved at 2 positions. The prodomains will be removed. the large subunit and the small subunit of two procaspase will be joined together to form one active caspase. So proteolytic cleavage of a procaspase results in activation. Once this gets started, this would be considered an initiator caspase, because this caspase will then proteolytically cleave other procaspases, resulting in this cascade of proteolytic cleavage, resulting in activation quickly, a number of different types. We're just naming them X and Y and Z. This could be, for example, SED4, which activates caspase SED3, which then activates other caspases. And SED3, for example, its job may be to cleave nuclear lamin. Another caspase that's activated, it may target a different cytoskeletal protein, right? Once caspase activity is initiated, quickly all other caspases are initiated, and they all have their activities initiated. They all have different roles targeting different proteins in the cell to quickly break down all of the structures to form these apoptotic bodies. So we'll leave it right here, and then we'll talk about how this is regulated, because we don't want all of our caspases activated all the time, and how this activity can be inhibited as well, right, to prevent caspases from being activated. So we'll pick up here on Wednesday and finish apoptosis, and then we'll talk about how new cells are induced to form as well. All right, let's see. Today is, what day of the week is it? Today's Wednesday, right? We'll continue this on Friday. I do have office hours today. I have office hours for you guys, a Zoom office hour from 1 to 2. If you have any questions, I'll see you then. Bye-bye.